Thank you for attending today's webinar, which is hosted by Mcule, and it's freely accessible for everyone. Let me start with a short introduction about myself. So my name is Murad Anwar, a custom solution engineer at Mcule, and I will be your host at today's webinar event. We will talk with our colleagues, friends from PostEra, an excellent company who aims to target cutting-edge solutions in drug discovery. And Aaron Morris, as the CEO of the company, will talk about their targets, their goals, including one of the largest COVID-related initiatives, which is called the COVID Moonshot 2020. Emil Ripka, the senior developer at PostEra, will share a demonstration with you. It will be about the brand new synthesis analysis tool, which is called PostEra Manifold. And Matt Robinson, the CTO at PostEra, will also attend during the Q&A session to help you answer your questions. So Mcule not just hosting this event, but also the part of PostEra's ecosystem. And from Mcule's side, Greg Prikler will share our solutions by building compound databases, sourcing compounds, and expanding the available chemical space. So just a few more words before I give the word to our speakers. In drug discovery, the long and costly process that is needed to discover a new drug is a never-ending challenge. So focusing to early drug discovery, our speakers here today are aiming to make this process cheaper and faster to enable you getting molecules easier than ever before. And now I'd like to give the word to Aaron Morris.
It's great. Thank you very much, Murad, and a particular thanks to everyone who's taken time out of your very busy schedules to come and listen to the work that PostEra and Mcule are developing. So I'm going to start here by giving you an overview of COVID Moonshot that Murad mentioned, as well as the Manifold product, which has really emerged as a result of our work doing COVID Moonshot. Emily is then going to give you a live demonstration of some of the features of Manifold. But firstly here, I'm going to share my screen and just quickly bring up an overview of both COVID Moonshot and Manifold. Give me a thumbs up, Greg, if that looks okay to you. Brilliant. So just for anyone who does not know about PostEra, is not aware of our company, in a nutshell, we focus squarely on medicinal chemistry. We focus on tackling the latest advances in machine learning that our team pioneers and applying that to the challenges that are inherent within the process of medicinal chemistry. As an organization, we're very proud that the machine learning that we use and that we offer to you as clients is not a black box. This is technology that has been validated, that has been published in top peer-reviewed journals, often in collaboration with some of our partners at large pharma companies. And so we really are very proud that you can have a look at how our technology is working and some of its key advantages as being the state of the art. We engage with clients in two different ways. The first way is by offering drug discovery as a service. We basically take our entire medicinal chemistry platform pertaining from finding hits to going from hit to leads and finally optimizing those leads. We work with some of the clients that are mentioned at this top row here, either to help co-develop preclinical assets or often co-develop technology that can then be applied broadly across internal drug discovery projects. We also work with some great biotechs where we focus much more squarely on problems in synthesis and hit expansion through a kind of manifold all-access collaboration where we really take some of the leading work that Manifold incorporates and apply it with a lot of the internal capabilities that PostEra has to rapidly shorten the cycle time. But as mentioned today, I really just want to hone in a little bit on COVID Moonshot. So COVID Moonshot was an initiative that PostEra helped launch and now leads about 9 months ago. And in a nutshell, the effort is to crowdsource a COVID antiviral, something that seems quite ambitious, but something that we've seen remarkable progress in. And I just want to walk you through very quickly how that progress has developed. So what was the origin story? Well, back in March of 2020, we saw a tweet that was actually put out by a team at Diamond Light Source who had run a fragment screen against the main protease, which is a key enzyme responsible for viral replication. And what they had effectively found were about 70 or 80 very promising hits that sat and bound nicely within the main protease. And this offered an exciting opportunity to look at fragment mergers to convert such fragments into more drug-like molecules. And so PostEra took this data and we put it on the web and we put it out in the open and invited scientists to contribute their ideas of how to begin to develop antiviral drug candidates. And to be honest, as a small company about 6 months old, we probably expected 50, maybe 100 submissions at best. We were pretty overwhelmed that now 9 months later, we've had well over 15,000 submissions from 400 individual scientists around the world. And more importantly than that, we've actually been joined by 30 global organizations who have taken these ideas, not just from merely designs on a website, but converted them into actual compounds that have been synthesized and then actual experimental assays being conducted. I think the need is still very, very clear that given that most antivirals will still be administered intravenously, given that these compounds are very kind of peptide-like, there's a great need for a small molecule orally bioavailable inhibitors of Mpro, particularly given the way that Mpro is relatively conserved across various strains of coronavirus. There's great potential for this being even a future pan-coronavirus inhibitor, so we think the need continues to be pressing. Here's just a snapshot, and I'm sure many of you who are in the audience here have been involved in some extent of the amazing organizations, and I think it's worth mentioning that all of these organizations have worked pro bono or at cost in order to take these design ideas and actually move them toward becoming hits, leads, and ultimately drug candidates. And we're immensely grateful for the synthesis, for the assays, for the computational support that has been provided to COVID Moonshot. Mcule in particular has been an invaluable partner in this entire process. And so really the aim of COVID Moonshot at the start was to move very quickly given the nature of the pandemic. And therefore PostEra's contribution was to do fast synthesis, was to find cheap, reliable chemistry that could move very, very quickly in terms of iterating on the cycles. And we were really, really proud to say that this, after 6 months, this slide is a 6-month snapshot after beginning COVID Moonshot, that we'd progressed from fragment hits to very selective, very potent leads in the nanomolar range. Not only that, but we then moved from biochemical assays into actual antiviral assays. You can see here that some of our lead compounds show comparable antiviral activity to remdesivir. And I think what was even more encouraging was that actually the lead compounds themselves were just very simple one-step amide coupling from building blocks that we found, again, been a real inspiration for the Manifold product. And so the lead compounds that we now have are going through in vivo testing, and the ambitious aim of COVID Moonshot is to have a candidate nominated for IND-enabling studies by March. This would be, as far as we are aware, even one of the first times, if not the first time in history, that a drug has been crowdsourced from scratch. So this is immensely exciting to us. However, these are some of the problems that we ran into that became the inspiration for developing the tool known as Manifold. Different contributors, as I mentioned, were given designs, and some have been helping us along the way. But we had one pharma contributor provide 16 compounds, and they sent these compounds to us saying that they should be put into the custom synthesis queue. They were specifically designed to be outside of any known virtual space. What we ultimately found was that of these 16 compounds, 9 of them could actually be purchased directly. And I think this really gave us a sense that even within large organizations, it seems that the ability to search these ever increasingly large virtual spaces is still a real challenge. Having a firm grasp on what is actually the live set of purchasable compounds is a very, very non-trivial problem, and something that really inspired our approach. Secondly, when we were doing synthesis with CROs, the actual cycle times themselves were really slowed down by just not knowing what inventory some of the CROs had at their disposal. And so, again, it was a real challenge of knowing if we are going to get this synthesis executed, which CRO is best placed in order to do it. And so, again, there was a real need within Moonshot to find a way to aggregate and get a dynamic, updated view of which CRO had which inventory and what were the lead times. And finally, though we've received 15,000 submissions, but they are by no means all synthesizable. And the majority of the contributors often were not able to find out what was the synthetic accessibility of their actual submissions. And so these problems really inspired the product that has become known as Manifold. And so just for the kind of final few minutes here, I just want to talk a little bit about what Manifold now offers you and some of the kind of specific advantages therein. So in a nutshell, as I mentioned, we take a really pragmatic approach to doing synthesis, particularly in the early stages where you're doing hit finding and hit expansion. We really don't think that trying to do 5 or 6 step synthesis early on is an optimal way to approach the drug discovery process. We seek to optimize for fast, reliable chemistry where you can get a very good set of feedback very quickly on the design hypothesis that you currently have. And then having received back some hits, be able to quickly find analogs and search around that chemical space that becomes promising. It's really critical then that in order to do that you have an updated inventory of what is purchasable. PostEra through Manifold has aggregated well over 10 billion purchasable compounds that we now can disconnect back to in our retrosynthesis and also offer similarity and search across. We also realize that there's a great need to be able to do rapid hit expansion, whether the hits you have are from fragment screens like with Moonshot, whether they're coming out of DNA-encoded library screens or high-throughput screens, the ability to quickly find analogs around that chemical space and identify where can I get them made or ideally purchased is again a very non-trivial problem that Manifold solves for you. And finally, we are huge fans of library synthesis. Being able to exploit the intermediates that exist within a variety of synthetic routes in order to quickly access a lot of chemical space is something that we've offered our clients that has saved them thousands of dollars and weeks of cycle time. And so just to point out a few specific features of Manifold that we think are a key advantage. Firstly, within synthesis, every single route that Manifold designs will always disconnect back to purchasable compounds. Not just purchasable compounds, but the largest set of purchasable compounds that exists. And this can give you confidence that you'll be able to access complex chemistry very, very quickly and often much cheaper than you would think. The routes that we actually send back to you through Manifold are prioritized both by synthetic success and by the actual strategic diversity that Emily's going to talk a little bit more about. We will not provide you hundreds of routes that you have to then filter through and try and find something that's viable. And finally, the actual inventory that we use is billions of available molecules. This is a huge engineering effort to have these updated and presented to you every single month. And I think this just really speaks ultimately to where we see the advantage of artificial intelligence and modern computational techniques. There's a big debate in the field as to whether a machine learning algorithm or a chemist can predict the outcome of a chemical reaction better. And though we find that debate particularly interesting, what we've ultimately realized is a machine will always outperform a human when it comes to remembering the millions and billions of building blocks that you can actually purchase and that your retrosynthesis ultimately disconnects back to. So we like to use computation and machine learning where they actually have a clear advantage and let chemists utilize their advantage. And secondly, the real value as well within Manifold is ability to do vast chemical searches. Both similarity and substructure, we offer 3 flavors of substructure, in fact, across well over 1 billion compounds, often in under a second. And actually exact search for your compounds in over 10 billion compounds. I think we're certainly aware that there's maybe a perception in the industry that that search and similarity are maybe solved problems, like aren't these things that we can already do? But actually, there are in organizations that can do some of these tasks to some extent. Often it's not that they have to work through the command line. Often the tools get outdated very quickly and the inventory becomes aged. The tools are often disparate, spread across different organizations. And so we wanted to bring all of these solutions under one house and into one product. And finally, we realized that ultimately delivering synthetic routes to you is of value, but in a world, particularly for biotechs, which are increasingly virtual, actually delivering molecules to you is what drives down cycle time. And so we actually offer, through PostEra, through Manifold, the ability to order compounds. We have all the existing relationships with great vendors like Mcule, with several CROs that we work with every single day. And so we realized that some organizations have a huge overhead of doing CRO management. This is a logistical way that we can also not only design the routes, but then get the compounds made and shipped and delivered to your doorstep. So ultimately, Manifold is there to help you get molecules, more than even just designing routes. And so how can you just quickly access Manifold? There are two different ways. So firstly, we have Manifold Pro. This will actually be available for you to sign up straight after this webinar. There is a free 30-day trial with no commitment. We don't ask for your credit card, and you'll be able to use the entire set of features that Emily is about to demo to you for those 30 days. We also have an enterprise version where we can deploy the entire Manifold infrastructure in a private cloud for you that is distinct from PostEra's infrastructure. On top of that, something that can also be immensely valuable for organizations is to actually customize the inventory within Manifold. Whether that's a function of the internal inventory that you have, or whether that's just a set of preferred suppliers that you would like to use, this is something we can offer through an enterprise version. And finally, there's a big gold padlock there because we realize that security is incredibly important. We realize that asking you to enter sensitive chemical structures into software is always something that should be approached with real prudence and with real sense of security around that software. And so PostEra's entire infrastructure, start to end, is HIPAA eligible. We don't retain any record of any of the structures that you enter into Manifold unless you specifically ask for those searches to be saved. We have spent an immense amount of time as an organization trying to give you the confidence that the highest levels of security are being used within Manifold itself. And here's just a quick summary of those two different versions outlining the various features that come with both of these offerings. And so just the final thing to end with on my side, PostEra, as I mentioned, covers the entire medicinal chemistry workflow. Manifold really sits in this area, which is useful for hit to lead around hit expansion. But if you are interested in broader drug discovery collaborations and services, please feel free to reach out to any of the founders at PostEra. We will get back to you incredibly quickly. But with that said, I'm going to hand over to Emily, who's going to give you a walkthrough of the Manifold tool. So thank you.
Great, Aaron. Thanks so much for that intro to Manifold. I'm just going to take the next 10 or 15 minutes of your time and use a couple of examples to demo the features that Manifold has to offer to hopefully get you guys as excited as we are to engage with the platform. So I'm just going to go ahead and share my screen here. Just give me maybe a thumbs up, Aaron, if you can see that guy. Okay, cool. So I'm going to go ahead and just pull up a Smiles from off screen here in a minute. But this is our main site, postera.ai. And what I'm going to be demoing to you is Manifold. So just postera.ai/Manifold. And when you go to this landing page, what you see is just our search query startup. So we have a bunch of different options for you to enter in your query. I'm just going to start off with a really simple example and just grab the Smiles over here and query it up for search. So when you search in Manifold, it's going to bring you to this initial loading page here, where currently what we're doing is we're searching for some routes using our retrosynthesis algorithms. And so you can see we're just grabbing some synthesis routes, and just in a couple of seconds, we have presented to you 15 synthetically accessible routes. So I'm going to go ahead and focus on a couple of the other tabs for this molecule. And in a future example, I'm going to really dive into the synthesis. So the first tab over from our synthesis is our exact match. So as Aaron was mentioning, we do almost instantaneous search of over 10 billion building blocks or molecules in order to find exact matches. And for our similarity and substructure, almost instantaneous as well. So in any vendor ID that you see on Manifold, so for example, here or back in our exact search tab, you can click on the link and it'll bring you right to the vendor site so you could purchase that molecule if it's just purchasable off the shelf or potentially made to order. So this one here is just an Mcule Ultimate molecule, purchasable under $200. So if we come back to Manifold, I'm just going to jump over to the next couple of tabs. So one over is our similarity search. And as Aaron was mentioning, you may think that like similarity and exact searching is all just like previously done, but we really aim to just bring it together in one location and have just a really coherent, fast, simple interface for you to really query and do some hit to lead expansion. So in the similarity search, if we can just go ahead and apply some filters, so I'm just going to look at Mcule only for the sake of this demo, but you can see that there's actually 13 similar molecules based on Tanimoto similarity. And any single molecule you see on PostEra's Manifold site, you can click through and it'll query up a new search for that specific molecule as well. So I'll just leave that guy loading in a new tab and just jump back to this starting molecule. But we also have a substructure tab after our similarity. And so we offer a couple different kinds of substructure. I'm just going to open it up again to all the vendors and maybe open up the lead time so we can see a lot of different options which are available here. So we have a superstructure subtab, which your query molecule will be included in these suggested superstructures. And then we also have Merkle scaffold. So we have Merkle scaffold, which takes into consideration the side chain positions, but also, or just doesn't, and it's just the scaffold. So we have 59 and 44 hits respective for that. And the last main tab on Manifold here is chemical properties and alerts. So again, we offer a lot of chemical properties for this query, as well as if there were any medicinal chemistry alerts, which we gather from the literature and from industry standards like Payne's filters, So you might have seen as well these export buttons. So on both the similarity and the substructure tabs, we have an export. So if you go ahead and click that, you can grab all of these physical properties and alerts for all of these potentially similarity or your substructure matches, and you can download it as a CSV file or an SDF. So I'm just going to pull it up quick to show an example here. But as you can see, when you pull up that document, you've got all of these chemical properties and alerts, as well as a vendor ID if you wanted to, in the future, after doing some maybe triaging of your own from using these properties, you could go out and purchase that building block. So I'm going to pull up another example quick, and this one is actually something which we have gleaned from the literature. So during our reading, we always try to find examples which we think would be helpful to test in Manifold to really demonstrate the idea of reducing human effort and costs through purchasing. So over to the right here, I hope you can see that double screen, but over to the right, this is just a scheme from the literature where the authors were really interested in synthesizing 4 different molecules. However, they were using 2- and 3-step routes, and we were really interested in just plugging these into Manifold and see if you could purchase these outright and see how our routes compared. So I'm going to use another feature for Manifold, which is just uploading a file. And I just have a text file with all 4 of these smiles here. And when you query up molecules, you could query 25 at a time. So I could just load this search up and then just press search. And it'll bring me to this search query result page where we have multiple different molecules which we want to search. And as you can see, all 4 of these are actually purchasable from multiple different vendors. And this page in and of itself could be just the endpoint to your search. If you were interested in just purchasing all 4 of these molecules, you could go ahead and you could export here as well, grab that CSV, and you have all of those chemical properties for all 4 of these guys, as well as the vendor IDs. I also want to dive into maybe an example of one of these. So I'm going to click on this third guy here and explore and see how our synthesis compares to the proposed synthesis that these authors used. So again, just pulling up this manifold query here, and we can see that we have 11 suggested synthesis routes. And if you scroll through, you can see that all of the routes are just one step. So they're really extremely simple syntheses. I mean, this isn't a very complex example, but just kind of illustrating that there. And you can also see that this top route underneath the exact match has the same intermediate building block that they were using for this actual two-step reaction. We just can purchase this guy off the shelf. I'll also point out that you can rank these routes by our strategic diversity and synthetic accessibility heuristics. However, if you jump down, you can also see that we also suggest the protected version of this molecule, which you could go ahead and deprotect. So I'm going to use one more example to illustrate some of our synthesis power. So again, just jumping back to Manifold here, and I'm going to grab a Smiles from off screen. So doing some DNA encoded library reading and research, we came across this molecule, and we're definitely interested in plugging it into Manifold to see what kind of routes were suggested for this synthesis. So again, just loading this guy into the search, and Manifold's going to go to work searching for some synthetic accessible routes to build this molecule. And while that's loading, I'm just going to bop over to the exact search for a second. You can see that some routes already popped up, but we'll come back once it's done loading. And you can see that we do have some database matches here. So these will be presented to you for if you have an exact match in the database literature for this molecule being synthesized previously. And another aspect I'll point out is this Save Search button here. So as Aaron was mentioning, security is really important to us. However, if you do want, and by default, we don't save any of your searches, but if you are interested in saving a search, you could go ahead and save the query, and you could open up your Save Searches tab on your account. And you have some saved searches there, which you can delete at any time. But I'll jump back to the synthesis here and re-rank these routes. And again, I'm just going to move it back to only Mcule just for the sake of the demo. And I'm going to open up the lead time to 12 weeks just because I'm really interested in seeing what routes are accessible at all, even if it takes maybe a little bit longer to get those building blocks. So the one aspect I haven't explored yet on Manifold is this Explore Route button. So again, we present 12 synthetic routes for this target, and you can scroll through and you can visualize all of the building blocks you would need to purchase in order to complete any of these syntheses. However, if you click this Explore Route button, we actually have another landing page for Manifold, which is just a visual tree representation of the route. And there's a couple of important points to note here. One, you can scroll through all of the results here, and you can see all of the different proposed strategies that we are suggesting. And you'll also notice some bond coloring here on the target molecule. And what that corresponds to is the different disconnections which are made for that specific route. And we really try to offer you a variety of strategic approaches so that you're not just using, so that we don't just present to you a bunch of routes with the same bond being broken, just with a slightly different, maybe halogen in a building block. So I'm gonna just scroll through these routes, and I'll stop on this guy quick. And I want to emphasize our references to you. So if any of these, if you wanted to explore the literature behind any of these reaction steps, you could go ahead and you could click on the reaction name. And what it'll do there is it will fetch some relevant references using some similarity algorithms from the patent databases. And so just wait for that guy to load up here for a second, and you can see that we're proposing, and then we have a bunch of different patent references over on the right. And you can click through any of these links, it'll bring you to the patent database. And the last thing I want to demonstrate to you are these buttons over here on the right. So we do offer you a way to export this single route. So if you really wanted to explore this route in particular, and you wanted to maybe use this strategy in your lab, we do offer this like nested representation of the reaction in specific with the vendor ID numbers so that you could go out and purchase those individual building blocks. And again, any single one of these building blocks that you see in this tree representation, you can also click through and it'll bring up a new query so that you could see if you could synthesize this guy if you didn't want to purchase it. We also have, lastly, a custom route designer. So we offer you a variety of synthetic strategies, and we think really accessible synthetic routes for even deep searches like this target molecule. However, if instead you wanted to go old school and kind of explore synthetic strategies, which you think you have a design for, but we haven't presented to you, you can in real time search the virtual chemical space. And I'll just pull up this guy quick and just make one disconnection here. And you can see that you can add the single disconnection and it will automatically populate the vendor ID numbers for a specific building block. So you could in real time potentially explore strategies which weren't suggested to you by us. And with that, I'll go back to our Manifold landing page here and note this button down here, need help, have feedback. We really appreciate feedback and we know there'll be lots of questions. So please reach out. We really would appreciate it and we'll get back to you soon. And with that, I think I'll hand it back over to Greg and we'll have some Q&A at the end where we can maybe dig into some questions.
Thank you, Emily, and Aaron for the demonstrations and presentation. So like you said, now I am going to give the words to Greg, but I would like to add also, I see there are a lot of questions arrived. So just remind you that at the end of the webinar, we will answer to these questions after Greg's presentation. So Greg will tell you more about Mcule's part in this and also about our solutions in getting molecules as quick as possible.
Thank you, Murad. Can everybody hear me well? That is great. Okay, so thank you, Murad, for the nice introduction, and thanks a lot, Aaron and Emily, for this presentation. It's always an honor to talk with you. I'm sure that this is something that very interesting and useful for many of you listening to this webinar, either in live or the recording. So I would really suggest you to take your time and look around in Manifold. I think, in my opinion, it's a fantastic tool. So let me share my screen with you quickly. So please let me know if you do see it. Can you see my screen now? Perfect. So in the next couple of minutes, I will tell you how and which of Mcule's tools and databases were included in Manifold, how we can help similar projects with the same amount of complexity. And we will also focus on our approaches in compound sourcing, focusing especially on our latest tool, SynthAgora, that is a platform for custom synthesis. So at Mcule, we support drug discovery by building and operating chemical marketplaces. We basically do this in two ways. We collect all available compounds and the relevant chemistry works around it, or relevant custom synthesis offers. And the second thing that we do is we expand the available chemical space for drug discovery. Our solutions can be represented in three layers: in-stock and virtual supplier compounds that are aggregated into the Mcule database, novel and synthetically accessible compounds from the Ultimate database, and our latest platform for custom synthesis, SynthAgora. Via our aggregated compound database, Mcule database, we collected 45 million unique structures. These are coming from 110 plus suppliers. Many of these are virtual, many of these are in stock. In fact, 9.3 million compounds are in stock ones, and these 9.3 million in stock compounds is the largest in stock chemical space from any other aggregator on the market. And these 9.3 million in stock structures, unique structures, is actually represented with 17 million supplier products. So these are represented with 17 million supplier products, and during the quote optimization, our algorithm finds you the best product combination to offer the best possible solution, usually offering fastest delivery time and the best price option as well. So including so many suppliers and so many products have obvious advantage on the chemical space because this is a large in-stock chemical space, but it also has an effect on the price of the compounds because in many cases we are able to find a cheaper product for a given structure of your interest. The second layer is the Ultimate database, which was developed in a 2 years EU-funded project together with trusted suppliers and pharma partners. This is based on currently 3 suppliers, 50,000 building blocks, and 170 reactions that are encoded. In this ecosystem, Mcule acts as a cheminformatic company who makes sure that the synthetic feasibility of the compounds are high and correct, and at least 80%, as we have developed our synthetic feasibility algorithm during the project. And I believe the development and the infrastructure of the Ultimate database would worth a separate webinar or presentation, but I will not go into details this time. But if you have any questions during the Q&A or later on, please feel very free to ask them. What is important to know now that is there are 125 million compounds that are only purchasable from the Ultimate database, can be delivered at known price, known delivery time with 80% synthetic success rate. And that's not a predicted number, but based on statistics, based on the last 10 months and also the development during the development project. And yeah, this is a database that, for example, Emily showed in her demonstration. So these are novel compounds that typically are more expensive than the in-stock one, but can add novelty to your screening deck. And the third layer is SynthAgora, which is our latest option and platform for custom synthesis. I'm quite sure that many of you have not heard about it yet, so I will go a little bit more into details about SynthAgora later on. Of course, in many cases, building databases is not enough because we need to give you additional support, supporting the full compound sourcing process during the way, so we can answer the usually complex request of pharma and biotech industry. And we doing so by allowing you to download and access both of our databases via API or just downloading it. That's how, and that's why it can be integrated into systems like Manifold. I think it's also important to highlight that we are the only aggregator company who built their full internal cheminformatic system. So we have built our full internal cheminformatic system, so we are not relying on any third-party tools or solutions. And this enables us to do quick development if needed for complex projects. So for example, we can develop custom API functions or share our database in a specific SD file format or the product level metadata. We also provide cheminformatic support if needed. We can help with compound selections or creating specific compound libraries for you. And as mentioned, by default, we do the quote optimization to find the best products for a given set or list of compounds, taking into account the price jumps in supplier price tables. Once the compounds are ordered, they are delivered to Mcule. We are able to store these compounds on your behalf, ship them later. We can do it in our HQ, where we also operate our that we use for reformatting. And with reformatting, we make sure that you actually receive the compounds in the format that you want them. So if you want them in plates or set of plates or in tubes, then that's the place where it happens. So to give you an example, let's say you order 10,000 compounds, 7,000 compounds might be from in-stock sources, 2,000, couple hundreds might be from the Ultimate, and a couple hundreds might come from custom synthesis. And regardless of the source, all of these compounds can be collected, plated, and put it in a set of plates or tubes and shipped to you in one single package. So finally, I would like to tell you more about our latest tool, SynthAgora. SynthAgora is a reverse custom synthesis auction site where you can submit the structures of your interest, and CROs are able to bid to those with price, delivery time, QC requirements, and other details in a unified and streamlined way, so it is easy for you to compare and select from them. once you selected the winning bid, you are also able to track the whole process, the whole ordering process within the same platform. And while suppliers are anonymous to each other, they anonymously able to see the winning bid and the winning delivery time for each project, so they can decide whether or not they are able to offer a cheaper or quicker option. Therefore, utilizing this competition, SynthAgora aims to reduce the two most important factors in custom synthesis: delivery time and cost. And we are very well aware that when we are talking about custom synthesis, IP does have a huge role in this, and we do our best to make sure that there can be no sensitive IP issue around this. So first and foremost, you are able to select the CROs that you want to expose your structures to within the platform. So you can select partners that you might already have an NDA with in place, and you might just using the platform to compare these offers. Secondly, we only allow CROs to register once they accepted our terms and conditions that are quite strict and basically act as three-way NDAs. They were developed by pharma partners, by legal team, and these three-way NDAs are acts between us, between you, and between the CROs to ensure that nothing can be exposed. And finally, we are also able to facilitate independent NDAs if needed. So the biggest CRO partners are already here and represented in the platform. PostEra is already represented here as a CRO. So while the Mcule and the Ultimate database are available at Manifold, you can access, or PostEra can see your request within SynthAgora if you select them. So that's again a nice back and forth integration in this way. So this is a fairly new platform, and I would be more than happy to give you just some insight on how it works, so you have a better picture. So if we go to synthagora.mcule.com, I think you should still see my screen. So you can log in quite simply with your email address and with the password. And once you did that, you are going to see the main page, your main page, where you can see all your auctions and all the orders. So you can track these, review these, and can make decisions if this is needed. So for example, in this case, we can see that there's an open auction here. We can click on the track auction option. Let me just zoom in just a bit to that. So we see that this auction is for these two structures. And in this case, we need 100 milligrams out of these. And in this current scenario, we have received bids from 5 CRO partners. Again, these are easily comparable data that you can see. So you might select the cheapest one or the quickest one, as all of them can meet your purity requirements. And you can, of course, look into more details about the bid. So we can click to bid details, and you can see, for example, whether or not a supplier accepted your exclusivity requirements, or the frequency of reporting, or what is the quality data or QC data they can provide. So you can see all of these. You can make your decisions and order a given compound. Once you did that, you can track the ordering process within SynthAgora. You are able to see that where the process stands. Is the supplier waiting for some building blocks, or they are looking in their in-stock collection for given building blocks? If they started the reactions itself, or is in the purification phase. And you are also able to see reports from the CROs within SynthAgora. Finally, let me show you how you can create an auction. So you just need to quickly click on the Create Auction button. I will not fill out everything because it might take a bit time, but you can name your auction. You can set an estimated budget or a goal budget and the delivery deadline. These are not mandatory, but definitely help the CROs to give you a bit that fits your needs or initial requirements. And again, you are able to select the CRO partners. You can select all of them or just a couple of them based on your previous requirements. Of course, if you find some CROs that should be presented but not, then please let us know by using this option here. We would do our best to include them. And then as a second step, you can select the compounds of interest. You can either draw a structure or you can paste multiple ones. Let me just quickly do that. In this case, I have pasted 4 compounds. I am able to add external IDs, and all of these external IDs would be following the compounds during the whole process. So you are always able to review them. So once you have these, in the third step, you are able to specify more specific requirements. The simple one like amount and purity, but also more complex one like whether or not you want to have a partial offer, for example. So in this case, we quoted 4 compounds, but we might know that these are quite complex, and we would be happy if we would receive a quote for at least 2 of them. So we accept partial offers. In that case, we are also able to set exclusivity or the frequency of reporting. And if we have, we are able to upload the synthesis route that also have the CRO. So for example, if we work with Manifold and we are able to extract the synthesis route, this can be a place where to use it. And if this is available, I would truly recommend it, because within this one single platform, within this one-stop shop, you are able to access so many CROs, and you can help all of them with this information and can hope for better pricing thanks to that. So with this, thank you from my end and from all of us here for your time visiting these webinars. We are more than happy to take questions. Thank you.
Thanks, Greg, for the excellent presentation and the demonstration as well. And as you said, now here comes the Q&A session. So here comes the first one, and it's for PostEra. So the question is from an anonymous attendee. So how is PostEra's synthesis approach differentiated from IBM Reaction, MIT, ASKSOS, and others?
Yeah, it's a great question. You're very aware of ASKOS and some of the great work that's been done by source solutions. So we actually have run head-to-heads on a list of about 100 chemical molecules. I can just show that slide really quickly, but I think what I would reiterate at a high level is that ultimately Manifold is there to help deliver molecules to you. So it absolutely does solve retrosynthesis problems. But in addition to actually finding out the best CROs to deliver your molecules, similarity search when you're looking for analogs, we really view it as broader than even just a pure retrosynthesis tool. But let me bring up the kind of like benchmark here, which I hope is somewhat informative. I think, Greg or Murad, would you mind just dropping your share screen so that I can share mine, if that's right?
Sure. So,
Appreciate
it's
it.
all
Thank
yours.
you. Thank you. Thank you. Okay, so we actually do have an appendix at the bottom of my presentation, which pertains to some of these head-to-heads that we ran. Okay, so this is 2 open source packages. In essence, Manifold does very well versus 2 of the more open source solutions that are out there, both in terms of the speed of execution and obviously the number of routes we can find. Certainly some of that will be down to the kind of more extensive building block inventory that exists within Manifold. So I hope that's a kind of helpful overview. I'm very happy to talk about kind of how the machine learning is differentiated, but in terms of the proof of the pudding, we do think that the way that Manifold constructs synthesis routes is competitive with other solutions that are out there.
Thanks, Aaron. Okay, and here comes the next question. If you can speak to how the compounds were chosen from among the 15,000 to synthesize and test versus MPro. And sorry for the name pronunciation. So the question is from Mcule+PostEra.
Sure, I'll take this one. Yeah, it kind of gets to the origins of Manifold, actually. So I think the first weekend we had 2,500 submissions, like right off the bat, and we FTEs ready to start at a couple different sites on Monday. So, we were rapidly spinning up inventories, using our tools, running them through. We also had a design prioritization. So, there's like hundreds of years of medchem experience also on the Manifold or the Moonshot, like wider team. And, we were having trouble getting everything to make sure the prioritized compounds met their preferences. So that's why we compiled like the thousands of chemical alerts to prioritize things. And so that first weekend, because we had multiple CROs starting, we had to, we were writing API calls to every virtual space, getting everything we could quickly, and then also figuring out what needed to be done, custom synthesis. So that was a lot of work from Alpha and I, and we joke about how long it took us compared to how quickly we could do it now with the tools we built. But it was caffeine, but it's still, we were able to do in a weekend what would have taken, just sending it off to CROs about a week or two. So
Thank you, Matt. So the next question is from an anonymous attendee, and it's for Mcule. So do you have any process through which you curate your vendors? One frustration you somewhat alluded to is ordering things that folks say are available but never show up.
Yeah, this is an excellent question. So yeah, there is two answers for this. So first of all, we curate the compounds that are not represented correctly. So we have a system called Mcule Advanced Curation that actually spots these incorrect representations. In a lot of cases, we did see some strange things represented in even bigger suppliers' catalogs. So we do curate those out. And the other thing is that when a company or a supplier say that they are available but they cannot be delivered anytime, so it's quite clear that these are rather virtual, then we set them with a different synthetic accessibility score, or that's stated that is from a virtual library, and we highlight that the delivery time should be 6 weeks of that. So first of all, we curate out the incorrect structure, and second of all, we have the daily communication with the suppliers, and based on statistics, we highlight our users what they can expect.
Thanks, Greg. So the next question is from Ji Shi. It's for PostEra. And the question is, what is the size of the reaction data used to train the AI retrosynthesis prediction models? And also, she had a question about the API, if it can be integrated. I think a lot of people asked about this, if Manifold can be integrated.
sure. So to the first question, our reaction database is, I think now around over 8 million, 9 million. And then we do a significant amount analyzing the reactions, cleaning them so that they work better in the machine learning pipelines. On the API axis, so yes, this is something that should be coming out quite soon on our end, especially should be very quick for people to integrate with similarity and substructure and exact searches, and then also on the retro alerts, and then on the retrosynthesis side, we'll probably be able to give you maybe number of steps. We can give you a route, but the trees aren't the easiest thing to read by eye. So we recommend once you get a route or see that the score is good, you then plug it into our system to view it. But yeah, that's on our roadmap to come out quite soon.
Thanks, Matt. The next question is also from an anonymous attendee. and it's for Mcule, about SynthAgora. What happens if a CRO quotes a 3-week lead time, but it takes 6 weeks until delivery? Are these CROs flagged for overly optimistic estimates?
That's another excellent question. Yes, they do. We plan to introduce shortly a scoring system where you are able to see how a synthesis partners they tell you the delivery time of the compounds and how often they are actually able to deliver the compounds. So there is a kind of a success rate that is presented for the synthesis partner. But I would also add to this question that a similar scoring is added to the customers as well. So if a customers request a lot of auctions but never actually orders, then they get a lower score because of that. So I would compare this to the system of Uber. So if the car is not clean and there's problem, then the driver will get a low rating. But if you do not show up for the ride, then you will also get a low rating. And that's important because in many cases, actually getting up with these bids and these offers are quite time consuming for our CROs. So it's important for them as well to see if the client on the other end is a reasonable one. So that's a both mutual scoring system, but that's a great and excellent question. Thank you.
Thanks, Greg. The following question will be from Dusan Petrovic, and it's the following. When you find a compound purchasable from multiple sources, like in your example, can you get an information on lead times from different vendors directly in Manifold? Maybe, Emily, it was in your demonstration already answered.
Yeah, so I guess I'll just answer that one quick. You can filter by lead time. So you could essentially just filter out any building blocks which you wanted sooner. We don't currently export the specific lead time, but you could apply filters to, I guess, get around that.
And here comes another PostEra related question. And thanks, Emily. So it's from Peter Stoughton. And the question is yield a parameter that can be used to rank the routes?
Yeah, it's a good question. So it's implicitly used right now in the score and the ranking you see. It's not a user option currently to be able to adjust by yield proof prediction of side reactions and things like that that could mess up a certain reaction. But it isn't currently like an adjustable parameter for the user. It's something we've considered and we're looking into, but it's not currently on there.
Thank you, Matt. Okay, so it's 7. Well, it's very late now, so we have time for one more question. It will be for Mcule. It's regarding the ultimate. So, how did you approach to maintain the synthetic feasibility for your chemical space?
Yeah, so regarding the ultimate database, to simplify this, synthetic feasibility algorithm behind the ultimate database is a set of filters and rules coded into smarts that are arranged in a complementary matrix. So the side and main reaction effects can be detected, and we can work with those. All of these are also complemented with AI-based approaches, and an enumerator is part of this as well. So it's rather complex tools. And during the development projects, we trained this in kind of iterative cycles with the supplier partners who did the reactions, and we trained this tool in every iteration and all the rules as well. I hope it answers the question.
Thank you, Greg. So I'd like to highlight that this recorded material will be available for you, and we will provide the video. So we will contact you via email about this information. And it was a very good experience, this webinar and your presentations. So let me thank you for all of you again, and I wish you a great day for the rest. Goodbye.